All right, let's jump into day 28. Today, we're taking a closer look at PDGF and VEGF receptors. You know, the ones. They're basically the brains behind wound healing and how our bodies build those brand new blood vessels.
It's really remarkable how these tiny proteins manage such intricate processes. I mean, they're essential for healing, of course, but they can also be hijacked in diseases, you know, like cancer, for example.
Yeah, it's kind of like a double-edged sword, isn't it? I mean, on the one hand, they're helping us recover, but on the other hand, they can contribute to tumor growth. Before we really get into the specifics of their signaling cascades, though, I think it might be helpful to step back for a second and look at the bigger picture of receptor diversity. I mean, it's easy to get lost in the nitty-gritty details if we don't appreciate all the different ways these cellular gatekeepers actually operate.
I agree. Sometimes seeing a concept in a bunch of different contexts really helps to solidify, our understanding. Why don't we take a quick detour to the world of glycine receptors? They offer a simple but really elegant example of how receptors can mediate cellular communication.
Glycine receptors. Now, hold on a second. Isn't glycine one of those amino acids, one of the building blocks of proteins? What's its connection to receptors?
Well, in the spinal cord, you see, glycine actually has a hidden talent. It acts as an inhibitory neurotransmitter. Think of it like the brakes on neuronal activity. It prevents things from getting too excited.
So instead of, building proteins, glycine is actually calming down neurons. That's quite a career change.
It is, and it achieves this calming effect by binding to its own dedicated receptor, the glycine receptor, of course. This binding triggers a change in the receptor's shape, which opens up a channel, and this allows ions to flow in or out of the neuron.
Oh, so it's like a gatekeeper then.
Mm,
It opens the door for specific ions to enter or exit.
precisely. And this flow of ions ultimately inhibits the neuron's activity, and this is how it keeps things balanced.
Okay, that makes sense, I guess.
Yeah.
Why are we talking about glycine receptors when we're supposed to be focusing on PDGF and VEGF?
Because it highlights a fundamental principle of how receptors function in general. They act like molecular switches, you see. They trigger specific cellular responses. When a ligand binds to them, in the case of glycine receptors, the ligand is glycine itself. But for PDGF and VEGF receptors, the ligands are PDGF and VEGF.
I see, I see. So even though the players are different
Yeah.
and the outcomes are different, the underlying mechanism of a ligand binding to a receptor and activating it is a common theme.
Exactly. And this concept applies to countless other receptors throughout the body. They all share this basic principle of recognizing specific signals and then triggering specific cellular responses.
Okay, that definitely helps put things in perspective. But before we go back to PDGF and VEGF, I'm kind of curious, are all receptors as straightforward as the glycine receptor? I mean, are they all just flipping between open and closed states?
Not necessarily. Some receptors can have more nuanced behaviors, like exhibiting different states of activation, or even inactivation. For example, the glycine receptor. It can enter what we call a desensitized state, where it becomes less responsive to glycine, even if glycine is still bound to it.
Oh, so it's like a dimmer switch with multiple settings.
Yeah.
Instead of just an on-off switch.
A perfect analogy. This ability to fine-tune receptor sensitivity adds another layer of complexity to cellular signaling. It allows cells to respond to signals with varying degrees of intensity and for different durations.
Wow, that's incredible. It seems like receptors are more than just simple gatekeepers then. They're like sophisticated control panels. They regulate cellular activity with remarkable precision.
Exactly. And understanding these nuances is crucial if we want to develop drugs that can target specific receptors
Yeah.
and, modulate their activity. Now, with that broader understanding of receptor diversity, let's shift our focus back to the stars of the show, PDGF and VEGF receptors.
All right, let's map out those cascades. But before we get lost in the molecular details, could you remind me why these two receptors are so important?
Well, think about what happens when you get a cut. Platelets rush to the scene right away. They form a clot to stop the bleeding, but they're also releasing a crucial signal, PDGF.
So PDGF is like a call for backup then. It's like it's alerting the body to the damage.
Precisely. PDGF acts like a distress signal. It summons cells like fibroblasts and smooth muscle cells to the site of the injury so they can repair the wound. It's the orchestrator of tissue regeneration.
And what about VEGF then? What role does it play in this whole healing process?
PEGF steps in when the body needs to build new blood vessels, so it can supply the healing tissue with oxygen and nutrients. It's the master architect of angiogenesis, the process of creating new blood vessels from existing ones.
Ah, that makes sense. So PDGF is about rebuilding tissue, and VEGF is about building the infrastructure to support that rebuilding. It's like a construction project with two specialized teams working in perfect sync.
A great analogy. Now, both PDGF and VEGF exert their effects by binding to their specific receptors, the PDGF receptor and the VEGF receptor, naturally.
Okay, so we've got these growth factors floating around, looking for their respective receptors. What happens when they find their match?
That's where the signaling magic begins. So when PDGF or VEGF, finds its receptor, it basically triggers a cascade of events inside the cell. And ultimately, this leads to, wound healing or angiogenesis. But what I find fascinating is that even though they have these different end goals in mind, they actually share some pretty striking similarities in their signaling mechanisms.
Oh, really? That's interesting. So even though they're tackling different tasks, they're using some of the same tools to do it.
Exactly. They're both what we call receptor tyrosine kinases. You can think of them as proteins with this built-in switch. That gets flipped on when the growth factor binds to it. And this switch activates what we call their kinase activity, which basically means that they can attach phosphate groups to other proteins.
So it's like adding a sticky note to a protein, and that changes how it behaves.
That's a great way to think about it. These phosphate groups act like molecular tags, you could say, and they can activate or deactivate proteins, triggering a chain reaction of events inside the cell.
Okay, I'm starting to get how these cascades work, but how does the cell know which pathway to activate? Or you've got wound healing on one hand and angiogenesis on the other. If both receptors are using similar mechanisms, how does it know which one to do?
Well, that's where the specificity comes in. The specificity of the ligands and the downstream signaling molecules. While both receptors are tyrosine kinases, they actually activate slightly different sets of proteins, which leads to those distinct cellular responses.
So it's like having two different sets of blueprints, even though the construction crew is using the same tools.
Exactly. Let's start with PDGF signaling, and we can kind of map out the steps involved in wound healing. So once PDGF binds to its receptor, the first thing that happens is what we call dimerization. That's when two receptor molecules come together, and they form this, dynamic duo.
So it's like they're joining forces
Yeah.
to make the signal stronger.
Precisely. This dimerization is crucial for activating their tyrosine kinase activity. And once they're activated, this receptor duo goes on a phosphorylation spree. They start tagging all these different proteins that are involved in things like cell growth, cell division, and cell migration.
And this whole phosphorylation cascade ultimately leads to the formation of new tissue to repair the wound.
That's the gist of it. One of the key pathways activated by PDGF is the PI3K-AKT pathway, which is a major player in promoting cell survival and growth. You can think of it like the fuel. That drives cell proliferation, making sure there are enough building blocks to repair the damage.
So PDGF triggers cell growth and division to patch up the wound. But what about cell migration? Don't those cells need to actually move to the site of the injury first?
You're absolutely right. PDGF also activates the MAPK pathway, which plays a crucial role in cell migration. You can think of that pathway as like a navigation system that guides the cells to the wound site.
So PDGF is like a two-pronged attack on the wound then.
Yeah.
On the one hand, it's stimulating cell growth, and on the other hand, it's guiding them to the right location. But how does the cell know whether to focus on proliferation or migration? I mean, do they happen at the same time, or is there some sort of switch that flips?
That's a great question. The balance between proliferation and migration is actually influenced by a bunch of different factors, like the specific type of PDGF molecule that's involved, the cell type that's expressing the receptor, and even the surrounding environment. It's a really dynamic process where the cell is constantly integrating signals to decide the best course of action.
Okay, so it's not just a simple on-off switch. It's more like a complex algorithm, taking all these different factors into account.
Exactly. Now, while PDGF is busy orchestrating tissue repair, VEGF is tackling a completely different challenge, building new blood vessels to support the whole healing process.
All right, let's switch gears then and explore the VEGF's signaling cascade. Does it also involve dimerization and tyrosine kinase activity?
Yep, the initial steps are pretty similar. VEGF binding also triggers receptor dimerization, and it activates that tyrosine kinase activity. But the downstream signaling pathways are different, which leads to the unique outcome of angiogenesis.
So it's like having two construction teams, using similar tools, but following different blueprints.
A perfect analogy. One of the key players in the VEGF pathway is a protein called PLC. Now, this enzyme cleaves a specific lipid molecule in the cell membrane, And this generates two really potent signaling molecules, IP3 and DAG.
IP3 and DAG, those names sound familiar. Don't they play roles in other signaling pathways too?
They do. They're like these versatile messengers that can activate various downstream targets. In the context of VEGF signaling, IP3 triggers the release of calcium ions from intracellular stores.
Calcium, isn't that involved in muscle contraction?
Yeah.
And a bunch of other cellular processing.
You got it. Calcium is like this universal signaling molecule. It's involved in countless cellular functions. In this particular case, the calcium influx that's triggered by IP3 activates a bunch of different proteins that are involved in cell migration, cell proliferation, and the formation of those new blood vessel structures.
So VEGF through PLCA and IP3 mobilizes calcium to drive angiogenesis. That's pretty amazing. What about DAG then? What role does it play in all of this?
DAG, on the other hand, activates another key enzyme called protein kinase C, or PKC for short.
PKC. Didn't we talk about PKA earlier? Is PKC like a relative of PKA or something?
They have similar names, but they're actually distinct enzymes with different roles. Both of them are kinases, meaning they can phosphorylate proteins, but they're activated by different signals. PKC, in the context of VEGF signaling, gets activated by DAG and calcium.
Okay, so different activators, different targets, different functions. It's all about specificity. What does PKC do once it's activated?
Well, PKC, kind of like PKA actually, is a busy bee. It phosphorylates a whole range of proteins that are involved in various cellular processes. In the context of VEGF signaling, PKC activation contributes to cell proliferation, cell survival, and the formation of those new blood vessel structures.
So we've got VEGF triggering PLCO, which then produces IP3 and DAG. IP3 releases calcium, which along with DAG activates PKC. And then PKC in turn phosphorylates proteins that drive angiogenesis. It's like a beautifully choreographed molecular ballet.
Exactly. And this intricate interplay of multiple signaling pathways really highlights the complexity of angiogenesis. It's not just a single pathway. It's a whole network of interconnected signals that orchestrate the formation of new blood vessels.
This is also fascinating. I'm really starting to appreciate the elegance and the complexity of these signaling cascades. But I can't help but wonder, if VEGF is so good at building new blood vessels, couldn't that be a problem in diseases like cancer?
That's an excellent question. And it leads us to the darker side of VEGF signaling.
You were saying something about a darker side to VEGF signaling. Is it that tumors can kind of hijack this process and use it to fuel their own growth?
That's exactly it. Tumors are like these demanding cities you see. They constantly need more and more resources just to be able to expand. And it's this insatiable hunger for nutrients and oxygen that drives them to manipulate VEGF signaling to their own advantage.
So instead of, helping to heal a wound, VEGF actually ends up building a superhighway for the tumor to grow.
That's a great way to put it. This process is what we call pathological angiogenesis, and it's a hallmark of many different types of cancer. It basically allows tumors to grow really rapidly and even spread to other parts of the body.
So I imagine that understanding the role of VEGF in cancer has opened up some new avenues for treatment.
Absolutely. This realization led to the development of what we call anti-angiogenic therapies. These therapies aim to starve tumors by cutting off their blood supply.
That's incredible. So how do these therapies actually work? Do they block VEGF itself, or do they interfere with its receptor somehow?
Yes. Well, both approaches are being explored. Some therapies use what we call monoclonal antibodies, which act like molecular decoys. They bind to VEGF, and that prevents it from interacting with its receptor.
So it's like intercepting the construction orders before they can even reach the building site.
Exactly. Other therapies use small molecule inhibitors that target the VEGF receptor's tyrosine kinase activity, essentially jamming the tools that are needed for blood vessel construction.
Fascinating. So by blocking either VEGF or its receptor, we can disrupt the tumor's blood supply and potentially even halt its growth. It seems like understanding these signaling pathways in such detail has given us some really powerful tools to fight cancer.
It really is a testament to the power of basic research. By unraveling the intricate details of cell signaling, we can identify vulnerabilities in these disease processes and then develop really targeted therapies.
This has been an amazing deep dive into the world of receptors. We started with a quick detour to kind of understand the basic principles of receptor function, using the example of glycine receptors.
Right. And then we mapped out the signaling cascades of PDGF and VEGF, two key players in wound healing and angiogenesis. We saw how they share some common mechanisms, like dimerization and tyrosine kinase activity. But they activate distinct pathways, leading to those very specific cellular outcomes.
And of course, we also explored how VEGF signaling can be hijacked by tumors to basically fuel their growth, which led to the development of anti-angiogenic therapies as a really promising approach to cancer treatment. It's amazing to see how understanding these tiny molecular players can have such a huge impact on our health and disease.
It really does highlight the interconnectedness of all these biological processes. And while we've covered a lot of ground today, we've only just scratched the surface of this vast and dynamic field. There's still so much we need to learn about how receptors orchestrate the complexities of life.
I feel like I've gained a whole new appreciation for the intricate molecular dances that are happening inside our cells all the time. It's clear that receptors are more than just simple gatekeepers. They're like these sophisticated control panels, regulating a wide range of biological processes with remarkable precision.
And as we continue to explore these signaling pathways, I have no doubt that we'll uncover even more fascinating insights that could lead to new treatments and a deeper understanding of ourselves.
It's a good reminder that even the smallest components of our bodies can have a profound impact on our health and well-being. Thanks for joining us on this deep dive. Until next time, keep exploring and keep learning.